Hey Bio 30s, I'm back again for another video. Get it? Because I walked in backwards. Anyway, we're going to be talking about sexual reproduction in terms of cell division, and that's going to be a main focus here on meiosis. So, if you look in your textbook, you'll notice that yes, we jumped over 17.2, which deals with cloning. We'll discuss cloning probably in our video tutorial sometime, as well as there may be a discussion question related to cloning as well. So I just want to jump ahead here to 17.3 and discuss meiosis. So last video we talked mitosis, cell division, one cell with 46 chromosomes, which we call diploid, becoming two cells with 46 chromosomes, also being diploid. Now, to start off here is the human life cycle. Now I use the term diploid and haploid, and let me explain what those two terms mean. So we have an adult male and an adult female. They are both diploid organisms. Diploid means two sets of chromosomes. So in this case, humans are 2N. The 2 refers to the number of sets. So we have two sets of chromosomes, right? One set of 23 from mom, one set of 23 from dad. So two sets. N refers to the number of chromosomes. So in this case, a human would have two sets of 23 chromosomes. Do a little quick math for you. Oops, too quick. We get a total of 46 chromosomes. So a human body cell would have 46 chromosomes. Why? Because you have two sets of 23 chromosomes. Now, when you undergo meiosis and produce gametes or sex cells, these are now haploid cells. And they're haploid, the sperm or the egg, because now they're just N. So we remember that a sperm contains 23 chromosomes and an egg contains 23 chromosomes. So now we refer to these cells as haploid cells. They're about half of a full set. If they were to come together through fertilization, which we've discussed, we form a zygote, which is now a diploid cell, 2N. This diploid cell will grow through mitosis because it is a diploid organism reproducing its cells through mitosis to make more diploid cells. Then we have this diploid baby here growing into an adult still through mitosis. When that adult or teenager, I suppose, hits puberty, they then have the ability to produce gametes or sex cells, which once again are haploid. So the difference between diploid and haploid is this, right? A haploid cell would contain just one set of chromosomes. In the case of humans, a haploid cell would be N equals 23. In a diploid cell, you have two sets of chromosomes. So if N equals 23, you have two sets, 23 from mom, 23 from dad, you get a total of 46 chromosomes. Okay, moving on here. Let's talk about mitosis. Or sorry, not mitosis, meiosis. Let's talk about meiosis. We already talked about mitosis. Comes in two stages, right? We actually have Meiosis I and Meiosis II. So we've actually two different divisions. All in all, we get 4 haploid cells, right? So in the case of a male, you get 4 sperm cells, 4 spermatids. In the case of a female, you get 1 oocyte or ovum, and 3 polar bodies, but still 4 sex cells, 4 gametes. Even though you only use one of them, you're still making 4 in total. So the first division is a reductive division. So through the process of Meiosis I, we're going to go from 46 chromosomes to 23. The second division is then simply taking those 2 cells that we've made, each with 23 chromosomes, and further dividing them into 4 cells with 23 chromosomes each. Okay, let's get into it. So, 23 chromosomes from the male, 23 chromosomes from the female, they match up and they find their pair. A pair of chromosomes is called a homologous pair, meaning they have similar information. Not exactly the same, because one chromosome might contain blue eyes and the other one might have brown eyes, but they at least have the information for eye color, okay? So they have similar information. They're similar in shape, size and gene arrangement, so the location of the traits, and they carry similar genes at similar locations, but once again, they are not identical. They're similar, but not exactly the same. So chromosome 1s would match up, chromosome 2s would match up, chromosome 3s would match up. Now, let's get into meiosis I. During prophase 1, just like in mitosis, the nuclear membrane will dissolve, freeing up the chromosomes. The chromosomes will condense, so going from that long uncondensed version to the condensed version. Spindle fibers would form, and here's the key difference between mitosis and meiosis, and I'll get my markers again to show you, okay? During mitosis, the nuclear membrane breaks down, and then we have 46 chromosomes kind of floating around there. During meiosis, the pairs, or the homologous pairs, one chromosome, let's say these are both chromosome 12, okay? This is chromosome 12 from dad, chromosome 12 from mom. They come together and form what's called a tetrad. So a combination of the two. So they come together and they form a tetrad. So a pairing of the homologous pairs. And you would never have chromosome 13 pairing with chromosome 21. That just wouldn't happen. The chromosomes would find its partner, its match, and form that tetrad or that pair. Now, during this pairing, a really cool thing can happen actually. So the chromosomes, and it's really hard to show with markers, I'll show in the video in a second, they kind of twist around each other. So the chromosomes will twist around each other. And what will happen is we have this process of crossing over where they intertwine, and sometimes bits and pieces can break off. And those bits and pieces can actually switch chromosomes. So the chromosome you got from dad, a little bit can break off and go on the chromosome you got from mom, and vice versa. And it's usually the information would swap at the same spots. So when things cross over, we get increased genetic variation, right? So we get increased variety. So you see a homologous pair here. They cross together and they curl around each other and they mix up, and then sometimes information can swap. So now these two chromosomes look different from where they started, and that gives us extra variation. This is why we can say, all right, mom's got blonde hair, dad's got blonde, well, I guess that's a bad example because it's recessive. Let's say mom's got brown hair, dad's got brown hair, and then the kid comes out with red hair, right? It's possible due to certain genetic inheritance patterns as well as the process of crossing over. So pretty cool process there. If I were to show you with the markers, it would look something like this. I would just switch the caps on the markers, right? Same genes crossing over, and now these two would look different than what we started. So now we have, right, our chromosomes, and they're a little different, right? We had some crossing over. Now, crossing over doesn't always happen, right? It's not a sure thing, but if it does happen, it's during prophase 1 of meiosis. There is zero crossing over in mitosis. So let's be clear on that. Next, we got metaphase 1. What's going to happen during metaphase 1? This is essential that you know this. During metaphase of mitosis, 46 chromosomes line up in the middle of the cell. Then we pull apart the chromatids. That doesn't happen in meiosis. In meiosis, the tetrads line up. So we are going to have, right, 23 tetrads lining up. Each tetrad contains 2 chromosomes. So now we have the tetrads lining across instead of just the singular chromosomes, okay? Then we move to anaphase 1. And instead of pulling apart the chromatids, we pull apart the tetrads. So that means one chromosome, a full double-stranded chromosome, is going over here, and then one full double-stranded chromosome is going over here. So now we have 23 over here and 23 over here. That's how we get our reductive division. That's how we cut the number of chromosomes in half. Really important to remember that segregation, or the law of segregation, will state that 1 to 23 goes over here, and then 1 to 23 goes over here. So you wouldn't have like chromosome 2 and 2 over here, and then this side would be missing a chromosome 2. So you get like 2 2s over there and 2 3s over there. It wouldn't work like that. You would want 1 to 23, one copy of each, and 1 to 23, one copy of each. That's anaphase 2. Remember, they're still double-stranded, so they're still double-stranded, okay? And then finally, we're going to finish off meiosis 1 with telophase 1 and cytokinesis 1, where now we're going to pull them apart, right? We pulled them apart, reform the nuclear membrane, and now we're going to split the cytoplasm. So at the end of meiosis I, we have two brand new cells. They do not contain the same number of chromosomes as the parent cell. They contain 23 chromosomes, right? And since there was crossing over, there's extra variation in there. So now these two new cells contain 23 chromosomes, and they're different from the cell that made them. In men, that'd be the spermatogonia and the spermatocytes, and in women, it'd be the oogonium and the oocytes. Now, let's finish up here with meiosis II. The nice thing about meiosis II is it's pretty much the same thing as mitosis, with one key difference. Obviously, we're dealing with 23 chromosomes instead of 46. So, we undergo prophase 2, and once again, this could be somewhat after meiosis I. So for sperm, meiosis II is going to be pretty quick after meiosis I. For the egg, you'll remember that the egg doesn't undergo meiosis II until it is fertilized. So it needs to wait for fertilization for meiosis II to occur. So prophase, nuclear membrane breaks down. Metaphase, now we have the chromosomes lying in the middle, and we're going to pull apart the chromatids. And this is going to happen in each cell. So we're going to pull apart the chromatids in each cell. Anaphase is the pulling apart of those chromatids. And finally, telophase, we make our nuclear membrane, and in cytokinesis, we split it apart. So at the end of the day, we end up with 4 cells, right? Cell 1, 2, 3, and 4. Each cell contains 23 chromosomes, so they're haploid cells. They're single-stranded, so they're not double-stranded chromosomes. And look, all 4 cells are different, right? All 4 of these markers are different. They contain a variation. So if you inherit this sperm, your eye color might be different if you inherited this sperm or egg or whatever we want to talk about. So this is how we make sex cells, and this is how we get variation within our gametes or our sex cells. Okay. One final kind of wrap up here to tie it all together, mitosis versus meiosis. I always like doing one of these comparison charts just to kind of show the difference between the two. So mitosis has one division, so it's just one prophase, metaphase, anaphase, telophase. Meiosis has 2, so prophase 1, prophase 2, metaphase 1, metaphase 2. Daughter cells are identical to parent cells in mitosis. They are different in meiosis. 2 cells are made in mitosis. 4 cells are made in meiosis. Remembering, in males, 4 spermatids, 4 sperm. In females, 1 ovum, and 3 polar bodies. Mitosis, we have a diploid cell making 2 diploid cells. For meiosis, 1 diploid cell making 4 haploid cells. We then have mitosis produces cells that are needed for growth and repair, new red blood cells, new white blood cells, new skin cells, things like that. Meiosis produces gametes or sex cells. No crossing over in mitosis, and of course there's crossing over in meiosis. So that's all I have for you today. Next lesson, we are going to talk about what happens if there's an issue with meiosis. So what happens if there's some type of abnormality? Like for example, what happens if these chromosomes of the tetrads don't split up? So that's called non-disjunction. So that's what we're going to talk about next lesson. So thanks again for watching, and we'll chat soon.